Good morning, good afternoon, and good evening to everyone tuning in from all over the world. Welcome to eLab Science Bioinnovations Global Webinar Series. It's great to see so many international participants joining us for today's session, and that is next generation CAR-T cell engineering for solid tumors. It will be overcoming the biological barriers and advancing the clinical translation. I am Dr. Sundar Qureshi, Global Scientific Liaison at eLab Science Bioinnovation, and I will be your host for today. During our time together here, we are going to talk through the key biological hurdles holding CAR-T therapy back when it comes to solid tumors. We'll also cover those next-gen engineering approaches closer to real-world clinical use. All right, so let's dive into the future of precision cellular immunotherapy. Before we dive into our topic, let me quickly tell you a little about eLab Science. We got our start back in 2009, and we have grown into worldwide life science company. Our products reach more than 150 countries, and we hold several key international certifications, including ISO 9001, ISO 13485, and CE. Our reagents have been featured in top-tier journals such as Nature, Cell, and Science. Back in 2011, we kicked off with basic biological research reagents and ELISA kits. Since then, our portfolio has kept growing. So now we offer antibodies, recombinant proteins, immunohistochemistry, Western blot kits, metabolism assays, food safety and animal disease kits, plus numerous flow cytometry antibodies. More recently, we have brought in some more advanced tools to our portfolio, things like APGplex, ELISpot, CellaQuant, and multiplex kit for magnetic-based immune cell separation. We also have received industry recognition, for example, CiteUp Supplier to Watch Award. And at the end of the day, all of this comes down to our focus on innovation and reliable research tools. Our goal is to support scientists, to support researchers so that you can design your experiment precisely, get trustworthy detection results, and make real research breakthroughs. Okay, before we begin, just few housekeeping notes. Your microphones will stay muted throughout the presentation. If you have any questions, submit them through the Q&A panel. We will answer them during the Q&A session. For the technical issues, please also use the chat box. A digital participation certificate will be provided to attendees who join the live session. Attendees will also have the opportunity to receive a complimentary eLab Science free sample. Details and claim instructions will be sent to your registration email. The webinar recording will be shared with all registered participants regardless of their attendance. So without further ado, let me introduce our today's speaker, Dr. Fahim Ahmad Khan. Dr. Khan is a senior researcher at Stem Cell and Cancer Research Indonesia with extensive expertise in stem cell biology, molecular immunology, cancer immunotherapy, and CRISPR-Cas9 genome engineering. His research focuses on advancing next-gen CAR-T cell therapies for solid tumors through innovative receptor design and tumor microenvironment modulation. He has authored more than 90 Scopus indexed publications, secured multiple competitive research grants, and actively leads international collaborations in translational cancer immunotherapy. We are honored to have Dr. Khan here with us today to share his expertise on next-gen CAR-T cell engineering for solid tumors. Dr. Khan, the floor is yours.
Thank you very much. Good morning, good evening, and good afternoon to everyone around the globe. So today, I'm going to discuss something about next generation CAR-T cell engineering for solid tumors. I'm Dr. Fahim Ahmad Khan, and I'm working as a senior researcher in Stem Cell and Cancer Research Indonesia. I'm also Associate Professor of Biomedical Science at Agung Putra University. So today, our main focus is on how we can tackle the solid tumors using CAR T cells. As we know, there are many problems regarding the solid tumors. The first problem we face is a heterogeneous antigen expression in the solid tumors. So to overcome this trouble, we have to have the CAR T engineered cells. In that case, the first problem we are facing is heterogeneous antigen expression. That comes from the inhibitory cells, the regulatory signals, and dense extracellular matrix that makes it the immunosuppressive tumor microenvironment. So the aim of the engineering is to decipher the TME, the tumor microenvironment, and the second we have to do the map antigen heterogeneity and immunosuppressive networks. Secondly, what we can do is using the synthetic biology to produce the Boolean logic gated receptors. In that case, we have FHCs, CFPs, and armored cars, which can also induce the payload delivery for improving the CAR T ability to target the solid tumors. Thirdly, we have the clinical translation and therapeutic index optimization, and that's what our objective is when we are using CAR Ts for the clinical application. As we know, in the hematopoietic cancers, the CAR Ts are very effective. However, when it comes to the solid tumors, the problem remains with the heterogeneity of the antigen expression. So when we are focusing on clinical translation, we have to make sure that the therapeutic index is the CAR T is having efficacy, quite high efficacy with low toxicity. So in that case, what we do is we actually navigate toxicity and safety measures and then use the predictive models to optimize the doses and therapeutic window. And also, we have to have the multi-omics integration pipeline. In that case, we use the genomics, proteomics, transcriptomics, and metabolomics in integration to find out relative markers which we can use for our targeting and improving the efficacy of CAR T cells. So in that case, the next generation horizons of multi-omics integration comes in, where we can engineer the sustained persistence, integrate multi-omics to refine memory T cell subsets and advanced platforms. So before going into the details, let's cover up some of the evolution of the cancer immunotherapy. So in early innovations, we all know we had the TILs. TILs are the tumor infiltrating lymphocytes. In that case, most of the time what we did in history was we used unmodified TILs with the high dose of IL-2, which established the curative potential of cellular transfer at that time. But the problem with the tails is remain the same, that the expansion capability of the tails was not uh, a lot, although its efficacy or its ability to enter the tumor microenvironment because of its specific antigen was there, but its use was limited because of its ability to expand, and also the ability to engineer those tails is also limited till date. Then in later years, we developed the technology, we call it CAR T. In CAR T, we have the synthetic T cell receptor that we call actually SCFP, and that is connected with a hinge, and it has some other domains like CD3 zeta or 41BB or CD28. So those were the engineering leaps when we come up with the CAR Ts. And the current frontier is next generation SynBio ECT. In that case, we call it adaptive cell therapy. So CAR Ts are also one part of the adaptive cell therapies. In that case, we actually move from the simple antigen recognition to complex synthetic biological circuits for solid tumors. Let's deconstruct the solid tumor microenvironment. And as we know, the solid tumor is posed with antigenic paucity and heterogeneity. So the scarcity of truly tumor-specific antigens and variable expression of tumor-associated antigens drive antigen escape and clonal outgrowth. So that is one of the constraints we have in the tumor hostile microenvironment. And the second barrier we have is a physical trafficking, where the T cells will face the dense extracellular matrix and also aberrant angiogenesis. So all those angiogenesis and aberrant extracellular matrix will cause the T cells not to enter the tumor microenvironment. And then the other barrier that we have is immunosuppressive cellular networks, where the regulatory T cells, MDSCs, or the T cell itself is causing that barrier. So high prevalence of Tregs, myeloid-derived suppressor cells, and tumor-associated macrophages blunt the effector function in that case. And the other thing is about the metabolic and soluble inhibitors. For example, the hypoxia conditions and the nutrient deprivation, and the extensive amounts of TGF beta IL-10 will provide the immunosuppressive environment where the T cells won't be able to enter the hostile tumor microenvironment. So to overcome these, we have some advanced receptor architecture. What we can do is, at the first, we have tandem and bicistronic CARs. In that case, co-expression of multiple SCFVs to target distinct TAAs, simultaneously preventing monoantigen escape. So in this way, we have multiple SCFVs, like the domains, the T cell receptors, synthetic T cell receptors, which can bind and target to distinct tumor antigens, and it will simultaneously prevent the monoantigen escape. So if you have noticed that when we are culturing it on a 2D culture, the antigens can easily be targeted with the cards, but when you are culturing it in 3D, because it has several antigens, so it can escape quite easily from the CAR T because it is designed for only CD19. Then we have the second way where the universal or adapter CARs come in, and in that case, a separation of the antigen binding domain from the signaling machinery using soluble adapter molecules, enabling programmable post-infusion target switching comes in. So these CARs can help help overcome the multi antigens to target. And then we have another strategy. We actually can do the T cell engagers, certain chemokines, cytokines will come in. So CAR T cells engineered to secrete bispecific engagers, redirecting endogenous bystander T cells against secondary TAAs to induce epitopes speeding. So in that case, what the CAR Ts will recognize is specific epitopes, and then it can target the tumors in quite strong ways. So let's overview the evolution of this CAR-T technology. In the beginning, as we see, we only had the endogenous TCRs, like T cell receptors. So that was the early phases where the lag cells and first generation TILs established the baseline. And then after that, we have the first generation of CARs where there was a SCFP domain, the synthetic TCR actually, which can recognize the CD19, and it was given a domain CD3 zeta. And in the second generation, we also added some of the co-stimulatory domains alongside the SCFP, and that is, for example, CD28. And then third generation or fourth generation also have that 4-1BB or IL-12 inducer. So as you know, IL-12 is also one of the very important interleukin that can actually keep the cells effective. So most of the time when we say that the CAR T cells are exhausted or not functional is because of those unavailability of the IL-12 or so then in the fifth generation of the CAR Ts, we also added another domain for IL-2 receptor. So it will keep the CAR Ts into its effective form. And later on, the addition of the ICAR, CCR, those inhibitory domains can help the CAR Ts to be effective only at the time when it is needed. So um, uh, in 2010, it was the second generation CARs, CD28, 41BB, co-stimulatory domains that create the CD19 paradigm and exceptional efficacy in B cell or an NHL helped with these targeted immunity with the second generation of the CARs. And then we had the third era where synthetic biology comes in, and it's continues from 2020s, and we are now in 2026. So in that case, bridging cell therapy, gene editing, and immune metabolism for solid tumors all come in one hand. So why this tumor bottleneck coming in is and why the adaptive adaptive cell therapy failures is because we have the locked door for example dysregulated chemokines. That means overactive WNT or beta catenin that will actually silence the T cell recruitment. And then we have antigenic heterogeneity, and that means polyclonal tumors trigger clonal escape and target downregulation. And another one is the immunosuppression environment of the tumor microenvironment. So exposure to the tumor microenvironment drives terminal T cell exhaustion. To overcome uh, this exhaustion, we use IL-7 and IL-15 as the other techniques, which are the interleukins, which actually restrict the T cells or the CAR T cells to its previous form, and it can get the maturation while in the body. So its exhaustion state is reduced by using IL-7 or IL-15. Uh, then we have logic gated circuits and CRISPR integration. So in that case, what we do is we can actually target the tox and the PDCD1. So in that case, the exhaustion drivers will be ablated, and that's how we can actually overcome the exhaustion of the T cells as well. So The first step is to ablate the multiplex, the tox and PDC1. So CRISPR-Cas9 will delete the inhibitory receptors like PD-1 and exhaustion drivers like TOX. And then the logic-gated circuits, what it can do is we can use AND or SYN-notch circuits that will ensure the T cells only execute killing programs upon dual antigen recognition. And then we have to see the safety profile that eradicates on-target off-tumor toxicity while enhancing polyfunctionality. So we should be very careful while we are, using that uh, s- logic-gated circuits because that can actually cause can can protect you from the cancer outcome. The second pillar in the CAR T is we can use the armored T cells and tumor microenvironment modulation. So if you see the dendritic cells and interleukins, if they are in a high concentration, for example, IL-12, this will be or M1 macrophage phenotype. It also come because of different kind of the cytokines that are released by the cells to keep those cells in the effective form, T effector cells. So those T effector cells can actually target the tumor in its in its bad form. So IF and gamma production or cell activation, cell survival will be high. Thirdly, what we can do is we all know there is a gut tumor axis. So in that case, if for example we can find out, like for example, Lactobacillus johnsoni or Clostridium sporogenes, those are actually producing the IPA, that is indole propionic acid. So we have found that actually indole propionic acid is responsible to keep the T cell in its effector form by interacting it with its epigenetic mechanisms. So those cells can keep the H3K27 acetylation form, and the T-pack stemness will be maintained. So microbiome driven adjuvants can also add up for the efficacy of the CAR T cells. So the rational engineering come in here is that live biotherapeutics maximize endogenous indole 3 propionic acid production, and systemic IPA maintains the H3K27 acetylation, sustaining epigenetic fitness in vivo. Let's discuss the Boolean logic gated systems for precision targeting. So the first challenge is the toxicity. In that case, the co-expression of TAs on a healthy tissue drives severe, sometimes fatal on-target, off-target toxicities. And how we can actually overcome is by inhibitory CARs, that is ICARs and not gate. So incorporate negative signaling domains, for example, PD-1, CTLA-4 directed at antigens exclusively expressed on healthy tissues, overriding activating signals. So the first thing first, SYN-notch binds antigen A on the tumor surface, and then the SYN-notch will releases the transcript factors, and then on third, when the transcription factor enters nuclear translocation and transcription of CAR-B will take place, and the CAR-B mRNA can later on binds the antigen B, triggering enzyme and perforin release. So in that case, the tumor cell will be recognized by the CAR Ts. The other strategy we have is armored cars. We also call trucks for the tumor microenvironment remodeling. In that case, we have the stroma degrading enzymes, for example, secretion of the ECM degrading enzymes facilitates the tumor parenchymal infiltration, and then paracrine cytokine deliveries can be localized, can be done. That is by localized secretion of IL-12, IL-15, or IL-18 upon CAR activation, counteracts local immunosuppression without the severe systemic toxicities of intravenous administration. And importantly, the checkpoint blockade integration is also important here. So in that case, co-secretion of anti-PD-1, PD-L1, and SEFPs, or integration of PD-1 dominant negative receptors share CAR T cells from exhaustion in both autocrine and paracrine manners. So the fifth generation of microenvironment responsive receptors is also dependent on hypoxia-inducible promoters. So utilization of hypoxia-inducible factor like if responsive elements restricts CAR or cytokine expression exclusively to the hypoxic core of solid tumors. And metabolic engineering, for example, overexpressing metabolic regulators enhances oxidative phosphorylation, maintaining effector function in glucose-dependent tumor microenvironments. And then the fifth generation card that we discussed previously as well, it actually can integrate intracellular cytokine receptor domains directly into the CAR construct alongside CD3 zeta and CD28, enabling antigen-dependent JAK state pathway activation. So when we are engineering, sometimes that JAK state pathway is not activated, which restricts its efficacy against the solid tumors as well. Ammonia is the other cause of that. Ammonia also makes the CARs ineffective in the uh, solid tumors. It activates the exhaustion of the T cells. So this graph actually shows the terminal exhaustion and epigenetic fixation. Our goal, mostly the terminal exhaustion burst comes up, but our goal is always to achieve stem-like T-Pax phenotype. So this is our goal, and when we are engineering our CAR Ts, we focus on the point that the stem-like T-Pax phenotypes should be sustained throughout the clinical use as well. So the first is the chronic antigen stimulation. So that means upregulation of inhibitory receptors, PD-1, TIM3, or LAG3. These are all the inhibitory receptors which are activated. And then in case if we get the stem-like T-Pax cells, so sustained function with lower inhibitory receptor expression will be the result for our experiments. So in this slide, what we try to show is the synthesis matrix where we have a certain kind of engineering solutions against the biological constraints. So as we know, we have antigen escape and heterogeneity as one of the constraints. What we can do to overcome this constraint is to produce tandem cars, universal adapters, TCE secreting cars. And the therapeutic rationale for this work is that targets multiple distinct antigens simultaneously, recruits bystander T cells to prevent monoantigen clonal outgrowths. On-target off-tumor toxicity is another biological constraint And the engineering solution that recent days have provided is the thin notch AND gates, ICAR NOT gates, or hypoxia responsive promoters. And it enforces strict spatial and temporal control over CAR expression, conforming cytotoxicity to the tumor bed. And another biological constraint we have is the immunosuppression that is provided by the Tregs, MDSCs, and TGF-beta or IL-10 as well. And then the engineering solution for that is we can have IL-12, IL-15 armored CARs with PD-1 DNRs and checkpoint blockade SCFPs. So what this strategy can provide you with is the repolarization local immune cells and actively shields the CAR T cell from checkpoint-mediated exclusion. We have the stroma like ECM, another physical exclusion constraint in uh, cancer biology. So solid tumors specifically. So ECM degrading CARs are heparinase can be used as an engineering solution for this. And how this therapeutic rationale works is because it can break down those physical barriers or bypasses systemic circulation to maximize local effector to target ratios. So let's discuss some of the preclinical innovation with clinical validation. So traditional preclinical models, as we discuss often, is a 2D monoculture. Why this is not working? Because in that case, only single, antigen can be targeted, and once we even validate our CARs in 2D models, it's usually fail in the real patient case because those cards will not be able to recognize the polyclonal nature of the solid tumor. So in that case, we have some advanced traditional models. So one of those first is to shift to 3D spheroids, organize and microfluidics to accurately assess T cell infiltration and ECM interactions. Secondly, we can have humanized in vivo models where utilization of HSPC humanized murine models to evaluate CAR T efficacy within a functional human innate and adaptive immune network can be used, and a rational delivery validation can be done in vivo as well by exploring intratumoral and intrapleural delivery to maximize local effector to target ratios while minimizing systemic exposure. And we have to be very careful with the toxicities. So in that case, standardized ASTS TCT grading and management of cytokine release syndrome and ICANS using IL-6 receptor and antagonists and corticoids can be used. So modulating SCFP binding affinity ensures CAR T cells that ignore the low density antigen expression of healthy tissue while remaining potent against high density tumor expression. The pharmacological reversibility for that is integration of inducible safety switches. For example, ICAS9 enables rapid apoptosis of engineered cells upon administration of an activating small molecule. So the frontier in vivo CAR T cell engineering is first bypassing um, ex vivo manufacturing, and then we also have targeted delivery vectors and transient versus stable expression. multi-omics and precision receptor design, we can use certain kind of genomic special transcriptomics and proteomics to find out our targets, the solid tumor targets that are possible with today's technology. And then this slide will show up to you the intelligent CAR T cell, which can actually address the heterogeneity, the microenvironment reprogramming, cellular fitness can be preserved, and we actually can bring up the paradigm shift from a single uh, antigen recognition to polyclonal antigen recognition by using certain kind of CAR cell therapies. And thank you for today and your attention. The keynotes you can take up with advancing specificity, enhancing safety, and we have to optimize the potency by overcoming barriers, and the paradigm shift is not a far cry from now on. So together we can engineer a future where cancer is no longer a barrier. Thank you very much once again, and I will give the floor to Dr. Sundas now. Thank you very much.
Thank you so much, Dr. Khan, for walking us through your thoughtful work on advanced cell therapy approaches for hard-to-treat cancers. If we want to move CAR-T research forward, we need dependable lab tools for isolating immune cells, checking cell phenotypes, confirming the therapeutic targets, and as well as measuring how cells functionally respond. Elab Science has a full range of products to help you along every step of your CAR-T work. That includes easy sort cell isolation kits, a wide range of flow cytometric antibodies, recombinant proteins, ELISA kits, plus the assays for cell health and metabolism. These different tools to work, you can isolate really pure T cell populations. You are able to check immune activation and exhaustion markers. You can explore tumor immune crosstalk, and you can measure cytokine output plus cell killing ability. This all helps us push forward better CAR-T option to treat solid tumors. All right, let's quickly walk through Elab Science product lineup, which is built for CAR-T work. This table here gives you a quick snapshot of our main product families, what they target, and which part of your CAR-T research they fit into. So let's look at some concrete examples here. For example, when we have, when we say we need to use the isolation, so we can have the easy sort kits for isolating CD4 positive, CD8 positive, or memory T cells. Flow panels to check exhaustion markers such as PD1 and LAG3. There are also several ELISA and ELISpot tools for cytokine measurement, plus cell metabolism and apoptosis assays. These are all practical reagents you can use across your CAR-T experiments. Now it's time for a question and answer segment. Please type your questions in Q&A panel, and we will address as many as possible. Dr. Fahim, I have a quick question here. Okay, someone asked, I have a problem which I encountered in my research. We generated CAR-T cells with a transduction efficiency of approximately 60%. And cells show excellent cytotoxicity against tumor cells in conventional 2D assays. However, when we evaluate them in 3D spheroids or organoid models, only a small fraction of CAR-T cells infiltrate the tumor core, resulting in poor tumor clearance. What experimental strategies would you recommend to improve CAR T infiltration without compromising cell viability or increasing off-target?
So yeah, this is a very interesting question, and actually it relates to improving the CAR T infiltration in a 3D model. So in that case, the key solutions he's asking, so what we can do is a chemokine receptor engineering. In that case, if we co-express receptors, for example, CR2, CCR2 for active homing along tumor chemical gradients, this will ultimately improve the the CAR T efficacy and infiltration in 3D models. So what he needs to do is he has to do the chemokine receptor engineering. Yeah, I hope it answers the question.
Okay, thank you so much. Okay, I just have another question here. Why is CAR T therapy more challenging for solid tumors compared with blood cancers? I think you have answered this question in your presentation,
Yeah,
but would you please elaborate a
yeah,
little bit?
exactly. So we actually facing several ECM problems. For example, the um, tumor microenvironment, suppressive tumor microenvironment that causes the CAR Ts to exhaust. For example, the availability of certain markers, specifically ammonia, is causing that the problem for the T cells to enter the CAR T specifically. So it loses its efficacy. Again, the PD-1 and all that access will come in, and IL-10 and IL-8 specifically will increase, or the other markers. So these problems and the polygenic nature of the solid tumor is responsible for CAR Ts not to be more effective. So that's why we have to engineer it and produce armor CARs with cytokine releases at specific time. So we have iCARs and all those things for that purpose. Yeah.
Okay. Next question is actually related to some essays from ELAB Science. So I'll answer this question. Let me read out the question first. Following CAR T and tumor cell co-culture, we see a reduction in tumor cell numbers, but we are not sure whether the cells are dying by apoptosis or another mechanism. Which assays would you recommend to confirm the type of cell death? Actually, I'd recommend combining an Action 5 apoptosis assay with caspase activity or tonal assay. Why? Because using more than one assay gives you a clearer picture of whether the target cells are undergoing early apoptosis, late apoptosis, or any other form of cell death that could even be ferroptosis or cryptosis. So next question, Dr. Khan, is during the repeated antigen simulation, our CAR T cells gradually lose proliferative capacity and show reduced IFN secretion. We also observed increased PD-1, TIM3, and LAG3 expressions after multiple rounds of stimulation. How can we determine whether these represents the true T cell exhaustion rather than temporary activation, and what approaches can restore a CAR T functionality?
Okay, so yeah, this is a solution-based question. So in my opinion, what you can do is the overexpression of protective transcription factors. For example, CJUN is one of the good transcription factor for that purpose, and you can also do the checkpoints. For example, checkpoint switch receptors, and those are PD-1, CD28 fusion, for example. If you do that, or optimized manufacturing with IL-7 or IL-15 will help you overcome this problem. So I'm just focusing on the solution. I'm not talking about the question originally he asked. So the solution is using IL-7, IL-15. Why I'm talking about that is because it will give the cell in a progenitor form, not the terminal, um, uh, you know, uh, terminal development. So it comes just before that. So when you use IL-7 as instead of IL-2, it will keep the cell in earlier form, and once you inject those CARs, it will mature in the body, within the body with the tumor antigens. So its efficacy will be higher.
Okay, thank you. Now, another question is related to the ELAB Science kits. Someone asked that they are quantifying IFN gamma, IL-2, TNF alpha, and IL-6 released after CAR T cell activation. But the cytokine concentration vary considerably between donors. And some samples fall below the detection range of our current ELISA kits. Which assay would you recommend for the sensitive cytokine analysis, and what strategies can improve assay accuracy and reproducibility? Actually, if your cytokine levels are falling below the detection limit, the higher chance is that the concentration of that particular cytokine target is truly low. So I'd recommend switching to a high sensitivity ELISA kit. For example, you can see for the samples with higher cytokine levels, a sandwich ELISA kit should work well. Also, you can check your sample dilution, which is very important, and make sure that your standard curve covers the expected concentration range. Those two factors often make the biggest difference in getting the reliable results. Okay, another question is, our selected target antigen is highly expressed on tumor tissue, but it also detected at low levels in several healthy tissues according to the transcriptomic databases, the public transcriptomic databases. So before moving into animal studies, how can we experimentally evaluate risk of on target of tumor toxicity? And are there engineering approaches that can improve tumor specificity?
So actually, we can implement the Boolean logic implement inhibitor CAR and that shut down activation upon binding healthy tissue markers. So the safety of switches will help in this case. Incorporate inducible suicide genes, for example, ICAS9, or reversible kinase inhibitors, for example, and like dasatinib, I think, to safely halt CAR-T activity in vivo. So this is how we can do the safety analysis.
Thank you. Another question is about the products again. Before CAR engineering, we isolate the primary human T cells from PBMC. Although the isolated cells achieved acceptable purity, we sometimes observed a reduced viability and lower lentiviral transduction efficiency after isolation. So which isolation strategy would be best? Actually, I have just mentioned in one of my slides that we are providing the magnetic isolation kits for immune cell isolation. So our kits use both the negative selection as well as the positive selection. For this particular CAR engineering, using negative selection is generally preferred because it enriches untouched T cells without directly labeling or activating the target population. Preserving the native phenotype and functional state of the cells typically result in higher viability, improved proliferation, and better viral transduction efficiency, and more consistent expansion through CAR-T manufacturing. We have also just launched a new brochure for CAR-T engineering. So if you are interested, you can always write to us. We can send you that brochure, and it will definitely help you a lot in your CAR-T research. Okay, all right, everyone. That brings us to the end of today's webinar. Thank you very much for being here and taking part in our session. We hope you picked up lots of useful takeaways from Dr. Han's talk, covering those evolving CAR-T engineering strategies, ways to get past tumor-related hurdles, how to improve the cell persistence and progress in precision immunotherapy for solid tumors. I'm sure that we couldn't get to answer all of your questions today. Even if you have any more questions, feel free to drop us an email at events@elabscience.com. Our team will get back to you with further support. Keep an eye on your registration email too. You will find the details there for webinar recording and extra related resources. We will also send the participation certificates and free sample claim form and the free sample list to all the attendees who have attended our session live. Thank you so much again. We hope that you will join us again for our upcoming webinars. Wish you all a great day and happy experimentation.